Hello, everyone. Dr. Nardi here. Welcome back to our exploration of behavioral neuroscience. Today, we are tackling one of the most remarkable feats of engineering in the human body, hearing. We are going to look at the auditory code. How does the biological machinery of your ear translate simple vibrations in the air into the rich psychological experience of music, speech, and noise? It is a journey from physics through biology and finally into psychology. Let's get started. Before we look at the ear, we must understand what it is detecting. As you can see on the screen, sound is not a thing. It is a process. It is alternating waves of pressure, compression, and rarefaction traveling through a medium like air. Think of ripples spreading across a pond when you drop a stone in it. We characterize these waves in two main ways, and these map directly onto our psychological experience. First, look at the top waveform. Frequency, measured in hertz, is how fast those waves are cycling. Psychologically, we experience this as pitch. High frequency equals high pitch. Second, look at the bottom waveform. Amplitude measured in decibels is the intensity or height of those waves. We experience this as loudness. The job of the auditory system is to capture these physical characteristics and translate them into neural signals. The process begins at the outer ear. Its primary job is collection and funneling. The part you see on the side of your head is the pinna. Its unique shape helps catch sound waves and funnel them into the auditory canal. These waves travel down the canal until they strike the tympanic membrane, commonly known as the eardrum. This is where air pressure is converted into mechanical movement. The eardrum vibrates back and forth, matching the frequency of the sound wave. On the other side of the eardrum, we enter the middle ear. This is an air-filled cavity containing the three smallest bones in your body, collectively called the ossicles. These are the malleus or hammer, the incus or anvil, and the stapes or stirrup. Why do we need tiny bones in our ears? The inner ear, where the actual processing happens, is filled with fluid. If sound waves in the air hit that fluid directly, 99% of the energy would bounce right off. Think of trying to talk to someone who is underwater while you're standing by the pool. The ossicles solve this problem through impedance matching. They act as a lever system, amplifying the force of the eardrum's vibration to efficiently transfer energy from the air into the fluid of the inner ear via the oval window. Now we arrive at the inner ear, housed within the incredibly hard temporal bone of the skull. The star of the show here is the cochlea, which looks like a snail shell. If we were to unroll that snail shell and take a cross section as shown in the diagram, you would see it is divided into three fluid-filled chambers. The scala vestibuli on top, the scala media in the middle, and the scala tympani on the bottom. The fluids here matter. The outer chambers contain perilymph, which is like cerebrospinal fluid. The middle chamber contains endolymph, which is unique because it has a very high concentration of potassium. That potassium is the battery that powers hearing. Zooming into that middle chamber, the scala media, we find the actual seat of hearing, the organ of Corti. This is where transduction happens, turning mechanical vibrations into electricity. The organ of Corti sits on top of a flexible floor called the basilar membrane. Sitting on that floor are the sensory receptors, the hair cells. We have a single row of inner hair cells, which are the primary reporters to the brain, and 3 rows of outer hair cells, which act as amplifiers On top of these hair cells are tiny hairs called stereocilia. As you can see in the diagram, these cilia are in contact with a stiff roof above them called the tectorial membrane. So, how does it work? This is the crucial moment of transduction. When the stapes pushes on the oval window, it creates pressure waves in the cochlear fluid. This causes the basilar membrane floor to bounce up and down. When the floor moves up, the hair cells are pushed against that stiff tectorial membrane roof. This forces the stereocilia to bend. Look at the close-up diagram. The tips of the cilia are connected by tiny springs called tip links. When the cilia bend, these springs stretch and physically pull open ion channels. Because the endolymph fluid is rich in potassium, potassium rushes into the hair cell. This depolarizes the cell, causing it to release neurotransmitters onto the auditory nerve fibers. Mechanical motion becomes a neural signal. We know that a sound occurred, but how does the brain know what pitch it was? For high frequencies, the brain uses place theory. The basilar membrane is not uniform. At the base near the oval window, it is narrow and stiff, like a tight guitar string. At the apex, or the far end, it is wide and floppy. Just like tuning forks, different parts of the membrane resonate at different frequencies. High frequencies vibrate the stiff base. Low frequencies vibrate the floppy apex. The brain knows the pitch based on where on the membrane the hair cells are firing. This creates a tonotopic map, a frequency map that is preserved all the way to the cortex. Place theory works great for high and medium tones, but the apex of the membrane is too floppy to distinguish very low frequencies accurately. For sounds below about 4 or 5 kilohertz, the brain uses temporal theory, specifically phase locking. As you can see in the diagram, auditory neurons fire action potentials at the same phase of the sound wave, usually the peak. If a sound is 100 hertz, the neuron fires 100 times per second. The firing rate of the neuron matches the frequency of the sound. For frequencies that are too fast for a single neuron to keep up with, groups of neurons work together in a volley principle to match the overall rate. Once the signals leave the cochlea via the spiral ganglion neurons, they enter the brainstem. The auditory pathway is complex, with several stops for processing. A helpful mnemonic used by some students is sonic hedgehog, but let's look at the actual anatomy. First, the signal hits the cochlear nucleus in the brainstem. From there, it travels to the superior olive, which is critical for sound localization. Next, it moves up to the inferior colliculus in the midbrain, which helps integrate auditory maps with visual maps. Finally, it reaches the medial geniculate nucleus, which is the auditory relay station in the thalamus. From the thalamus, the signal is projected to the cortex. The destination is the primary auditory cortex located in the superior temporal lobe, specifically on Heschel's gyrus. Crucially, look at the color coding on the brain diagram. The tonotopic organization established in the cochlea is preserved here. Neurons that respond to low frequencies, shown in red, are grouped together. Neurons that respond to high frequencies, shown in blue, are also grouped together. The cortex is organized into columns, processing not just frequency, but also comparing input from both ears. Besides what the sound is, we need to know where it is. The brain figures this out by comparing the signals from the two ears. For low frequencies, we use interaural time difference. If a sound comes from the right, it hits the right ear a fraction of a millisecond before the left ear. In the brainstem, specifically the medial superior olive, we have neurons acting as coincidence detectors. They only fire strongly when signals from both ears arrive simultaneously. Based on which neuron in the array fires, the brain computes the time delay and determines the location. For high frequencies, time differences don't work as well because the sound waves are too short. Instead, we use interaural level difference. High frequency sounds bounce off the head, creating an acoustic shadow on the far side. If a high-pitched sound comes from the right, it will sound louder in the right ear than the left. Neurons in the lateral superior olive compare these intensity differences to determine location. Unfortunately, this delicate machinery can break down. We generally categorize hearing loss into two main types. The first is called conduction deafness. This is essentially a mechanical issue in the outer or middle ear. It happens when sound acts physically blocked from reaching the cochlea. Common causes include simple wax buildup, a punctured eardrum, or ossicles that have become calcified and will not vibrate. The good news is that these issues can often be fixed medically or helped with standard hearing aids that simply amplify the sound. The second type is sensorineural deafness. This is a problem located deeper in the inner ear. It usually involves permanent damage to the hair cells themselves or to the auditory nerve. We often see this result from loud noise exposure, aging, or the side effects of certain drugs. Crucially, in mammals, once auditory hair cells die, they do not regenerate. The loss is permanent. However, for severe sensorineural hearing loss, where the hair cells are gone but the nerve is intact, we have incredible technology, the Cochlear implant. This device bypasses the damaged hair cells entirely. It uses a microphone to pick up sound and a processor to break it into frequency bands. It then sends electrical signals down a long electrode array threaded directly inside the cochlea. Look at how the electrode is placed. It takes advantage of place theory. It stimulates the base of the cochlea for high frequencies and the apex for low frequencies, directly activating the spiral ganglion neurons and restoring a form of hearing to the brain. To summarize our journey today, we started with physics, pressure waves in the air. We moved through biology, the mechanical systems of the outer and middle ear, and the electrochemical transduction in the organ of Corti. And we ended with psychology, how the brain interprets those neural codes as pitch, loudness, and location. The auditory system is a masterclass in biological translation, turning the vibration of the world around us into the perception of sound. I will see you in the next lecture. Please like, subscribe, and share this video.